The human nervous system spent approximately 300,000 years calibrated to a specific environment. Grasslands, forests, rivers, open sky, the sounds of wind and water and animals. The sensory inputs of the natural world were not background to human life. They were the entire context in which human biology evolved. The indoor, screen-mediated, acoustically artificial environment that most people in developed countries now inhabit for the majority of their waking hours is, from the perspective of the nervous system, an experiment with no evolutionary precedent. And the research of the last 2 decades has been measuring what that experiment costs in cortisol levels, in immune function, in blood pressure, and in the measurable biological responses that occur when people return to natural environments even briefly. 20 minutes in a natural environment produces measurable reductions in cortisol, not reductions that require self-report or mood questionnaires. Reductions measurable in saliva samples, in blood pressure cuffs, in heart rate variability monitors. The body responds to natural environments through mechanisms that do not require conscious awareness or deliberate relaxation practice. They are automatic, physiological, and rapid. This is the story of what those mechanisms are, what the Japanese research on forest bathing has established about immune function, and why the 20-minute dose that produces these effects is accessible to almost everyone. Richard Louv introduced the term nature deficit disorder in his 2005 book Last Child in the Woods, a phrase describing not a clinical diagnosis but a pattern of observable consequences from the progressive disconnection of modern life from natural environments. Children spending less time outdoors, adults working in artificially lit spaces for the majority of their days, urbanization concentrating more of the world's population in environments where natural stimuli are absent or muted. The concept captured something that researchers had been documenting independently, that measurable biological markers of stress, inflammation, and immune suppression are higher in populations with limited access to natural environments, and that exposure to nature produces biological changes that are not explained by exercise or social interaction alone. The research separating nature's biological effects from exercise and social contact has been methodologically careful about this distinction. Studies comparing walks in urban environments to walks in natural environments, matched for duration, distance, and social context, consistently find that the natural environment produces greater reductions in cortisol, greater reductions in rumination, greater improvements in heart rate variability, and greater reductions in activity in the subgenual prefrontal cortex, the brain region associated with repetitive negative thinking, than the urban environment. The difference is not effort, it is environment. Something in the natural environment produces biological effects that the urban environment does not, and identifying what that something is has occupied researchers across multiple disciplines. The autonomic nervous system is the primary pathway through which natural environments produce their biological effects. The autonomic nervous system has 2 branches, the sympathetic, which drives the stress response and mobilizes the body for action, and the parasympathetic, which drives recovery, digestion, and restoration. Modern urban environments, characterized by noise, artificial light, visual complexity, social density, and the cognitive demands of screen-mediated life, maintain a chronic background activation of the sympathetic branch. Not the acute sympathetic activation of a threat response, but a sustained low-level arousal that keeps cortisol mildly elevated, keeps heart rate slightly above the resting baseline, and reduces the variability in heart rate that indicates healthy parasympathetic tone. Natural environments contain what environmental psychologists call soft fascination, sensory inputs that are interesting enough to engage attention without requiring the directed cognitive effort that urban environments demand. Flowing water, wind in leaves, the movement of clouds, birdsong, these inputs hold attention gently, without the sustained cognitive load of traffic, screens, notifications, and the social complexity of dense urban interaction. This shift from directed cognitive effort to soft fascination allows the default mode network, the brain's internally focused processing system, to settle into a different mode of activity. Rumination, which involves the default mode network generating repetitive negative self-referential thoughts, requires some degree of directed cognitive engagement to sustain. Soft fascination displaces this engagement without replacing it with another cognitive demand. The rumination cannot maintain itself. The parasympathetic nervous system activates as sympathetic tone declines. Heart rate variability increases. The spacing between heartbeats becomes more variable, which is paradoxically the signature of a healthy, responsive autonomic nervous system. cortisol begins to fall, blood pressure decreases, the body shifts from a sustained alert state into a recovery state. A study published in Frontiers in Psychology measured cortisol in adults before and after 20-minute periods in either natural or urban settings. The natural setting produced significantly greater cortisol reduction, approximately 21% greater reduction than the urban setting within the 20-minute window. 20 minutes, measurable in saliva, no meditation required, no deliberate relaxation practice. The environment produced the effect. The Japanese research on Shinrin-yoku, forest bathing, a practice formalized by the Japanese Ministry of Agriculture, Forestry and Fisheries in 1982, has extended the biological documentation of nature's effects beyond the autonomic nervous system into immune function, specifically natural killer cell activity. Natural killer cells, NK cells, are a component of the innate immune system that identifies and destroys cells that have become cancerous or infected with viruses. NK cell activity is a measure of immune surveillance capacity, the immune system's ability to detect and eliminate aberrant cells before they can establish disease. NK cell activity declines with age and is suppressed by chronic stress, chronic sleep deprivation, and chronic psychological distress. Ching Li, a researcher at the Nippon Medical School in Tokyo, conducted a series of studies measuring NK cell activity in working adults before and after forest bathing trips, typically 2 to 3 days spent in forested environments with walking and exposure to forest air. The findings were consistent and substantial. After a 3-day forest bathing trip, NK cell activity increased by approximately 50% compared to pre-trip levels. This increase persisted for more than 30 days after the trip, not returning to pre-trip baseline until approximately a month had passed. A single 3-day exposure to forest environment produced immune benefits that lasted a month. Li's subsequent research identified the likely mechanism. Forest air contains phytoncides, organic compounds released by trees and plants as part of their own immune and defense systems. Alpha-pinene, beta-pinene, limonene, and other terpenes that give forests their characteristic smell are biologically active in the human immune system. When inhaled, phytoncides stimulate the production and activity of NK cells through mechanisms involving reduced stress hormones and direct effects on NK cell gene expression. Li conducted a controlled experiment in which participants spent nights in hotel rooms with or without wood essential oils, a surrogate for forest phytoncides, diffused into the air. The phytoncide exposure group showed significant increases in NK cell activity and significant reductions in adrenaline levels compared to the control group without any nature exposure. The phytoncide compounds themselves, inhaled during sleep in an urban environment, produced measurable immune effects. This finding is significant because it suggests that the immune effects of forest bathing are not entirely dependent on the visual, auditory, and cognitive aspects of the natural environment. The chemical compounds in forest air are themselves biologically active, and their effects on NK cells are independent of the relaxation and autonomic responses that natural environments also produce. Blood pressure is the third biological variable with consistent documentation in nature exposure research. Hypertension, elevated blood pressure, affects approximately half of adults in the United States and is a primary risk factor for stroke, heart disease, and kidney disease. The relationship between chronic stress, sympathetic nervous system activation, and elevated blood pressure is well established. Sustained sympathetic tone increases peripheral vascular resistance and cardiac output, maintaining blood pressure above the optimal range. Studies of regular Shinrin-yoku practice in hypertensive adults have found clinically meaningful reductions in blood pressure following nature exposure programs. A meta-analysis of randomized controlled trials examining nature-based interventions found that forest bathing produced average reductions of approximately 1.4 mmHg systolic and 1.5 mmHg diastolic, modest but consistent reductions that, at the population level, would correspond to meaningful reductions in cardiovascular event rates. For people with borderline or mildly elevated blood pressure, regular nature exposure, combined with other lifestyle interventions, represents a genuine non-pharmacological contribution to blood pressure management. The blood pressure effects appear to require regularity rather than single exposures for sustained benefit. A single forest walk produces acute blood pressure reduction that returns to baseline within hours to days. Regular weekly nature exposure, consistent with the practice of Shinrin-yoku as a weekly or bi-weekly activity, produces sustained reductions that are maintained between sessions. The minimum effective dose is the practically important question because most people cannot arrange 3-day forest bathing trips on a monthly schedule, and the research suggests that meaningful benefits are achievable with much more modest exposure. The Michigan study published in Frontiers in Psychology established that 20 minutes in a natural setting produced measurable cortisol reduction. The research on attention restoration, the recovery of directed attention capacity through exposure to soft fascination environments, suggests that benefits begin within minutes and plateau at approximately 20 to 30 minutes for most people. For the immune effects documented in the Shinrin-yoku research, longer exposure appears to produce more substantial and longer-lasting NK cell increases. The 3-day trips that produced 50% NK cell increases and month-long persistence are at the high end of what most people can implement. But Lee's research also found that shorter one-day nature walks produced NK cell increases that were detectable for at least a week, smaller in magnitude, but meaningful in duration. The practical recommendation that emerges from the available evidence is approximately 20 to 30 minutes in a natural environment, park, forest, riverside, or any green space with trees and natural sounds, at least 2 to 3 times per week for the sustained autonomic and cognitive benefits. For the immune effects, one longer nature exposure per month, a few hours to a full day in a forested environment appears to produce benefits that bridge the gap between shorter weekly sessions. Natural environments do not need to be wilderness. Urban parks with mature trees, riverside paths, and green spaces with natural sound sources produce measurable effects. The key variables appear to be the presence of vegetation, natural soundscapes, reduced urban noise, and ideally tree cover, which is the primary source of phytoncides. Here is the synthesis. The human nervous system evolved in natural environments over 300,000 years. The modern indoor, urban, screen-mediated environment is an evolutionary novelty for which the autonomic nervous system has no calibration. The result is chronic sympathetic activation, mildly elevated cortisol, reduced heart rate variability, sustained cognitive load, that natural environments rapidly and reliably reverse through the mechanism of soft fascination and reduced directed cognitive demand. 20 minutes in nature produces measurable cortisol reduction. The biology fills in why it matters. The environment that produced your nervous system is also the environment that most efficiently restores it. 20 minutes. Trees. Natural sounds. If this changed how you think about the green space near your home or workplace, hit the like button. Share it with someone who spends their breaks looking at a phone screen instead of going outside.